Good morning, all of you. Now, today I am going to start a new topic from Unit 3rd. Topic is that B part है Structures and Function of MHC. Structures and Function of MHC. M for major, H for histocompatible, C for complex. Okay, let's start. In the slides, introduction, definition, history, types of MHC molecules, structures of MHC molecules, HLA, H2 complex, functions of MHC molecules, ये सारी चीज़ें हम यहाँ पर डिस्कस करेंगे. Now, introduction part, you can see here major histocompatibility complex. You can see here histocompatibility है, it should be histocompatible. Both term word is used somewhere. So major histocompatibility complex is a set of surface proteins located on the cell membrane of nucleated cell. Means we can say these are surface protein which is present on the plasma membrane on WBC and other nuclear cells that are unique for each person except identical twins. So MSC are surface antigens present on cell surface. These antigens produced from the plasma membrane into extracellular fluids. These are cell identify markers are unique for each persons except identical twins. Now it plays more important work to identify the antigens between self and non-self body, intracellular recognitions and responsible for antigens presentation. Histo, histo means tissue. Compatibility means refers to living together harmoniously. Okay. MSC molecules always recognize only T lymphocytes. The two types of MSC are what in immunity? T helper cell, which is represented by TH, recognized by MSC molecule second, and T cytotoxic cells are recognized by MSC first molecules. This T helper cell and cytotoxic cells is responsible for producing antibodies. Now you can see in next slide definitions of MSC. Major histocompatibility complex is a membrane attached protein which works on recognition of antigens between self and non-self body and antigen presentation. Now history, you can see here Peter Gorer in 1930 found that 4 group of MSC molecules. He used the blood samples of mice to identify the blood groups antigens which designed by first to fourth group of MSC. George Snell, Jean Dosset and Berry received a Nobel Prize in 1980 for the contribution to the discovery of MSC molecules. Now here we can see classification of MSC molecules. These are 4 types: class 1st MSC, class 2nd MSC molecules, class 3rd and class 4th. Okay. Now in this 4 classification of MSC, 1st and 2nd are very important. Now class 1st MSC molecules. Class 1st MSC 45 kilodaltons molecules are a group of major histocompatibility antigens They are present on the surface of all nucleated cells except nervous tissue and platelets. It presents antigens to TC cells. It binds with CD8 adhesion molecules of TC cells. TC cells, cytotoxic cells. It brings about cell-mediated immune response. And one thing you should be note down on RBC, cell MHC cell is not present because on RBC there is a blood group antigens present. Okay, now class first MHC are glycoproteins in nature and transmembrane molecules present on all nucleated cell and encoded by HLA and HLB and HLA-C genes. It consists of two polypeptide chains, alpha chains and beta 2 microglobulins. You can see here consists of two polypeptide chains, alpha and beta 2 globulin. Alpha chain which is no covalently attached with beta microglobulins, बहुत strongly bind नहीं होते हैं. Alpha chains contains a transmembrane glycoproteins which is encoded by A, B and C genes of grouped HLA. Okay. Alpha chain is organized by three domains such as alpha 1, alpha 2 and alpha 3, each domain containing 90 amino acid sequences. Next point, beta 2 microglobulin is similar in size of alpha 3 and it does not contain transmembrane proteins. And next point, when the antigen is internalized and processed inside by proteosomes, the peptides are produced. Peptides is further loaded on the group of MHC first molecules from endoplasmic reticulum. You can see here class first MHC antigens diagram. Alpha 1 and alpha 2, there is a peptide binding group, and membrane distal domains, this is alpha 3 structures, this is a transmembrane segment, this is the membrane proximal domains. Okay, these are the structures of class first MHC antigens. You can see here alpha 1 and alpha 2 in human beings at chromosome number 6. These are found at alpha 1 and alpha 2, and this is also binding site. Okay, this group is for a binding sites. Then you can see alpha 1 and alpha 2. In case of class first MHC antigens, there is a binding site in between alpha 1 and alpha 2. Okay, now in class second MHC molecules are present on the surface of antigen presenting cell and cell which engulf the foreign antigens. It binds with exogenous endocytic degradations antigens. It binds with CD4 adhesion molecules TS cells. It also consists of two polypeptide chains, namely alpha chains and beta chains. Alpha chains and beta chains. Antigen is processed inside the endosomes and peptides is further loaded on a group of MHC second molecules. Now next point, the class of MHC molecules consists of two polypeptide chains, namely alpha chains and beta chains. In alpha chains 33 kilodaltons and beta chain is 28 kilodalton chains. The both chains are attached non-covalently. Each chains contain 2 units. The 2 units of alpha chains are called alpha 1 and alpha 2. And the 2 domains of beta chains are called beta 1 and beta 2. Beta 2 and alpha 2. Beta 2 and alpha 2 are transmembrane domains anchoring the MHC2 plasma membrane. The alpha 1 and beta 1 domains joint bear a peptide binding group. You can see here alpha 1 and beta 2 jointly bear a peptide binding group. मतलब between alpha 1 and beta 2 there is a space that is antigen binding site space. Okay, in previous means class first MHC you can see the space where antigens binding, that space is different. You can see here between alpha 1 and alpha 2 there is a peptide binding site, but in MHC second molecules the space for antigen binding site is in between alpha 1 and beta 1. Okay, this is the main difference between MHC first and MHC second molecules. Okay, you can see here alpha 1 and alpha 1 and beta 1 there is a space that is a binding group, peptide binding group, and this MHC second is responsible for provoke immune system distribution is specifically up to limited cell, and this MHC second molecules help in killing pathogens. Okay, now class third MHC molecules. The molecules include complements like C2 and C4 and BF factor B, and class 4th MHC molecules. These molecules is present on T cells of leukemias as well as on immature thymocytes. तो ये था अबाउट MHC के बारे में. तो बेसिक जो इसमें फर्स्ट और सेकंड है इन्हीं का मेन रोल है, क्योंकि ये जो MHC फर्स्ट और सेकंड है ये जो है एंटीबॉडीज़ प्रोड्यूस करती हैं और ये जो है हेल्प करती हैं टू फाइट या कंबाइंड विथ या बाइंड विथ एंटीजेंस. ओके, तो डिफरेंस बिटवीन फर्स्ट एंड सेकंड MHC molecules you should be know with respect to their binding site, कहाँ पे वो बाइंड कर रहे हैं और उनका रोल क्या है. ओके, तो हमने देखा कि जो बाइंडिंग साइट है MHC फर्स्ट में वो आपने यहाँ पे देखा दैट इज़ अल्फ़ा 1 एंड अल्फ़ा 2. ठीक है, तो अल्फ़ा 1 और अल्फ़ा 2 में ये बाइंड करते हैं, फिर जो है कि यहाँ पे जो पॉलीपेप्टाइड्स हैं वो सरफेस पे आ जाते हैं, जैसे पैथोजेनिक एंटीजेंस इस पे बाइंड करते हैं. और and MHC first also involved in graft rejection and cell mediated cytolysis. Okay, so you should be know the differences between MHC first and MHC second. Now there are some terms and points which are included in these slides are HLA. तो you should be know what is the HLA. तो HLA क्या है? H for human, L for leukocyte and antigens, human leukocyte antigens. तो HLA is the human leukocyte antigen. HLA is the MHC molecules present in human beings. HLA is a set of surface proteins present on the surface of all nucleated cells. They are responsible for graft rejection, adaptive immunity, defense against infection. Sometimes it is expressed on the cancer cells, destruction, certain autoimmune diseases, and the certain complements. Okay, these are the specific uses of HLA. MSC is the general terms referring to the cell surface antigens of vertebrates. So as we know that in human beings at chromosome number 6 genes of MSC प्रेजेंट एट इन शॉर्ट आर्म. जो क्रोमोजोम है, एक क्रोमोजोम के जो दो पार्ट हैं, एक बिगर आर्म क्रोमोजोम है, ठीक है, इसी को हम HLA कॉम्प्लेक्स बोलते हैं, और एक है शॉर्ट आर्म क्रोमोजोम्स. ओके, नाउ S2 कॉम्प्लेक्स. S2 कॉम्प्लेक्स ऑफ माउस. देखिए अभी हम लोगों ने जो HLA कॉम्प्लेक्स पढ़ा वो ह्यूमन बींग्स के क्रोमोजोम नंबर 6 के जीन्स पर जो प्रेजेंट होता है उसके रिस्पेक्ट में था. लेकिन ये जो माउस या माइस में है, ये इसका क्रोमोजोम नंबर जो है वो बदल जाएगा, तो क्रोमोजोम नंबर 17 है. ठीक है, तो इन माइस एट क्रोमोजोम नंबर 17 इज टर्म एज S2 complex. So the major histocompatibility complex of mouse is called S2 complex. Okay, S2 complex is a cluster of genes responsible for the production of antigens located on of nucleated cells and complement components. This complex is located in the short arm of the chromosome number 17. Okay. Now next point. It consists of a set of structures genes. The genes that make up a given histocompatibility complex are called halotypes. Now, functions of MHC molecules. MHC molecules are loaded with a bit of sample peptide fragments derived from degradation of protein present inside the cell. This peptide is the mirror image of proteins, mirror image of proteins present inside the cell. MHC molecules contain self as well as non-self antigens, and they bring about defense against the infections and diseases. They mediate certain autoimmune diseases. They are responsible for individual smell of people. Now, come on summary. The both MHC and MHC first and second molecules are responsible for antigen presentation, and it has applications of antigens recognition between self and non-self recognition. Mostly they are located on T lymphocytes encoded by chromosome 6 of the human. The two types of antigens, degraded peptides, exogenous and endogenous, are involved to complete this process of antigens neutralization. Okay. Now these are references. तो ये था about structures and function of MHC. Thank you all of you.